Welcome to the EvoEd COVID-19 video series. In this video, we'll take a look at the Delta variant. We'll discuss its evolution and then look at what might allow it to cause reinfection in hosts. By the end of this video, you should be able to explain the concept of a host pathogen arms race as it relates to the evolution of Delta, and be able to describe the L452 mutation and explain how it makes Delta different than previous variants of the virus. The Delta variant was first detected in India in late 2020, but wasn't declared a variant of concern until May 6, 2021. The major causes for concern were how quickly the variant was spreading and that it was causing reinfection in those who were infected by a previous strain of the virus, as well as breakthrough infections in those who were fully vaccinated. So, what allowed the Delta variant to reinfect individuals with prior immunity? The short answer is evolution. The long answer is a process known as a host pathogen arms race. As hosts within a population become infected by a viral pathogen, they begin to produce antibodies, which can protect them from future infections from the same virus. This means that viruses can no longer effectively replicate within the host and spread between these immune individuals. And any variant of the virus that has the ability to evade the existing immunity of hosts will replicate more frequently and grow in numbers. It's called an arms race because the process resembles a military arms race, where two opposing sides continuously aim to outdo the other. Where a military arms race may result in the production of more and more arsenal, a host pathogen arms race involves the continuous progression of strategies to remain immune for the host and strategies to evade this immunity for the pathogen. Here's another way to visualize the outcome of a host pathogen arms race. Antagonistic refers to the fact that the interests of the host and pathogen oppose one another, and CoAdaptation refers to the fact that the adaptation in the virus causes adaptation in the host, and vice versa. An important thing to note here is we are dealing with two completely different types of adaptation. The virus is adapting through natural selection. In other words, variants with more effective tactics outcompete others, and the alleles that underlie these tactics increase in frequency within the population. On the other hand, in this example, we're talking about adaptation in the context of changes in the host's immune system. This is not the same as adaptive evolution because we aren't dealing with a change in the allele frequency of hosts. With that being said, a similar process would be observed if we looked at changes in the frequency of phenotypic characteristics in a host population over many generations in response to a pathogen. This is called antagonistic coevolution, but of course, this type of observation isn't possible within the scope of the current pandemic. So in the case of Delta, mutations that allowed the virus to evade previous immunity played a big role in its rapid spread around the globe. But what specific mutation drove Delta from being able to evade the adaptive immunity of its hosts? Well, there are likely a few that play a role, but one that has been specifically implicated is the L452R mutation that results in a change on the receptor binding domain of the spike protein. L452R means that amino acid 452 is changed from a leucine to an arginine. So that red dot in the red box represents a single amino acid changed at that location on the protein. Now, let's take a look at the specific mutation that leads to this change. Here's a small portion of the genome of the original SARS-CoV-2 virus. The nucleotide triplet highlighted in blue codes for the leucine at amino acid position 452. Since we're dealing with RNA, this would actually be expressed as CUG in the virus. In order for this amino acid to change from a leucine to arginine, all that's required is a single nucleotide substitution that changes the middle uracil into a guanine, resulting in a missense mutation. What does this seemingly small change mean for the function of the virus? Well, the difference in chemical structure slightly changes the shape of the receptor binding domain of the spike protein. And the host's antibodies are proteins that are structured in a very specific way that allow them to bind to the invader like a key fitting a lock. This binding neutralizes the virus by preventing it from binding to ACE2 receptors. the antibodies that were produced in response to the spike protein from a wild-type virus during a previous infection can bind to the binding domains of similarly shaped spike proteins and help prevent reinfection. However, the single amino acid change from leucine to arginine changes the shape of the binding domain enough so that some of these antibodies can no longer bind to it. While the wild-type virus will be quickly dealt with by the immune system, the host will need to produce new antibodies that can bind to the changed spike proteins. By then, the virus will have already had time to replicate within the host and potentially spread to new individuals, leading to the establishment of new strains of the virus. So that's the story of Delta and host pathogen arms races. Does an arms race mean the pandemic will never come to an end since new variants will continuously evolve? Not necessarily. In the story of Omicron, we'll discuss how evolutionary factors might actually help bring the pandemic to an end. Well, that's all for this one, and we'll see you next time.